So welcome back, guys. USMLE Step 1 2025. USMLE 2025 Step 1 Immunity, or better said, immunology. So we're gonna read now about natural killer cells. So the natural killer cells are a type of innate immune cells of lymphoid origin. They being part of the innate immune system. So the natural killer cells recognize and kill unhealthy cells by looking for non-specific abnormalities. So the natural killer cells are stimulated by interleukin-2 and interleukin-12. Natural killer cells are stimulated by interleukin-2 and interleukin-12, and interferon beta and alpha, and interferon alpha and beta. So the natural killer cells, their function, they enhance their function by attacking or killing cells lacking MHC1. So any cell that doesn't show up in any MHC1 is going to be attacked by natural killers. So which is down, the MHC1 is down expressed by virally infected and cancerous cells. So if you have a cell that is infected with cancer or turning into cancer, or another cell that is virally infected, they down express the MHC1, and then the natural killers can notice that and attack those cells trying to kill it. So the natural killer cells can also attack cells that are bound to immunoglobulin G antibodies in what is called antibody-dependent cell-mediated cytotoxicity. In order to kill a target cell, natural killer cells release perforin and gramzyme. So they release perforin and gramzyme. Just remember that. That's the way the natural killers they can target or kill the cells. They release perforin and gramzyme. The perforin open holes in the target cell's membrane, which gramzyme enter to induce cell death. Okay? So remember, natural killer cells are they are lymphocytes of the innate immune system. They get stimulated by 4 things. Number 1, interleukin-12 and interleukin-2. Interleukin-12, and they're stimulated by interferon alpha and beta. They produce interferon gamma, and they are activated no specific surface signals. So they are activated by no specific surface signals, like cells lacking of MHC1. So if they see any cells that is lacking MHC1, they attack them, and that's in the case of cancer cells, malignant cells, and viral infected cells. They produce perforin and gramzyme. The perforin make holes in the cell membrane, and the gramzyme get into and produce or induce cells death. Okay, remember that. Let's see. So coming back here to the first aid 2025, this is for step 1. The major function of the natural killer cells, or they are lymphocyte member in the innate immune system, use perforin and gramzymes to induce apoptosis of virality infected cells or tumor cells. We just learned that. So activated and housed by interleukin-2 and interleukin-12, and interferon alpha and interferon beta. They produce interferon gamma. Remember, they produce interferon gamma, and they are activated by interferon alpha, beta, and the interleukin-2 and 12. Remember, this is important, that the interferon gamma that the natural killer produce it activates the macrophage. It produces activation of the macrophage. They turn the macrophage into an incredible Hulk. So into the incredible Hulk, they transform the interferon gamma produced by the natural killer, transform the macrophage into the incredible Hulk. So the macrophage, that he can engorge bacteria, but when it's under the effect of interferon gamma, they turn into a killing machine. And just remember, they are induced to kill when they notice any cells that doesn't show up MHC1 on their surface. There's another mechanism that they can kill, is also kill via antibody-dependent cells-mediated cytotoxicity by the union of CD16 bind to Fc region in the immunoglobulin G. So let's jump now into the major function of B and T cells. B cells, humoral immunity. So remember, B cells, humoral immunity, recognize and present antigen, undergo somatic hypermutation to optimize antigen specificity, produce antibody. They differentiate into plasma cells to secrete a specific immunoglobulin. Remember, most of the immunoglobulin come from B cells. Maintain immunology memory. So the memory B cells persist and accelerate the future response to antigen. So that's the main function of the B cells. They recognize and present antigen. They produce antibodies and immunoglobulin, and they maintain immunology memory. So let's see the T cells. The T cells are part of the cell-mediated immunity. Cells-mediated immunity. B cells are humoral immunity, and T cells are cells-mediated immunity. So remember, we have two kind of T cells, CD4 and CD8. CD4 T cells they're helper T cells. They help B cells to make antibodies and produce cytokines to recruit phagocytes and activate other leukocytes. That's CD4. And CD8 T cells, they are directed kill virus infected and tumor cells via perforin and gram-symes. That's shown like we know about natural killers. So they act similar to the natural killer cells. CD8 T cells, they directly kills virus infected and tumor cells via perforin and gram-symes, similar to natural killers. The type 4 hypersensitivity reaction, they are responsible for the type 4 hypersensitivity reaction. So the T cells are present in hypersensitivity reaction type 4. Hypersensitivity reaction type 4 related with T cells. So they are responsible for acute and chronic cellular organ rejection. Acute and chronic cellular organ rejection, T cells. So remember, the house of the T cells is the thymus. So they go to the thymus to develop and differentiate. So in the thymus, there's a group of process that it's called positive selection, negative selection. This happen in the thymus, and then the T cells have to go there to being released to the blood or to be killed, because if they have too much self immunity, they attack each other. They have to be destroyed. We're gonna explain it here. So in this process, positive selection and negative selection. So the positive selection, this happens in the thymic cortex. Keeps the T cells that recognize self-peptides to allow the cooperations in immune response. It's double positive thymocytes expressing TCR that recognize self-peptide MHC complex receive a survival signal. So in this process, you're gonna see better explained in the graphic we're gonna show in a little bit. But this is very high yield. I've seen questions about this. When this is failing, that's why some people have like a immunology disease, like a hyperreaction to your own blood, things like that. So negative selection, that happened in a thymic medulla. Remember, the other part, positive selection, is in thymic cortex, and then negative selection is a thymic medulla. In this process, remove T cells that bind too strongly to self-peptides. So if they bind too strongly to the same peptides, it's not good, because you have immune response, or like a immune allergies to the skin, things like that. Thymocytes expressing TCR with high affinity to self-antigens undergo apoptosis of death, or become regulatory T cells. The immune regulator IIR proteins drives negative selection, and deficiency lead to autoimmune polyendocrine syndrome. This is a chronic mucocutaneous candidiasis, or hypoparathyroidism, and adrenal insufficiency and recurrent candida infection. This is a mnemonic that says, without air your body will chair. Without air your body will chair. So chair is a group of disease that could happen if you don't have this negative selection working perfectly. So the autoimmune regulator IR protein drives negative selection. And deficiency look to autoimmune polyendocrine syndrome. So if this negative selection doesn't happen right, you're going to have this group of disease very likely. So remember, the autoimmune regulator protein drive negative selection. The autoimmune regulator IR, this protein drives negative selection. If this negative selection doesn't work right, doesn't work perfectly, then you very likely is going to have one of those 4 diseases. So the 4 diseases that could happen if you don't have a correct negative selection are chronic mucocutaneous candidiasis, hypoparathyroidism, chronic mucocutaneous candidiasis, hypoparathyroidism, adrenal insufficiency, and recurrent candida infection. This group is, the mnemonic is going to be chair, like a sitting on a chair, like sitting on a chair. Chronic mucocutaneous candidiasis, recurrent candida infections. It's the same thing. I would say it's the same thing, so it's easy to remember. Chronic mucocutaneous candidiasis in the mucocutaneous candidiasis, recurrent candida infection, hypoparathyroidism, and adrenal insufficiency. If the negative selection doesn't work right, you're going to be very likely infected or affected with those diseases. So that's what this part of negative selection is high year. So let's keep going. Before keep going, let's check the graphic here. So this is a differentiation of T cells. Remember, the T cells precursors are positive for CD4 and CD8 at the same time. That happened in the cortex here. In the cortex, You can see in the thymus here. So in the cortex of the thymus, you're gonna have the positive selection. So in this case here, they are CD4 and CD8 positive. We're talking about the T cells. Before they come here to the thymus, they was born in a bone marrow. They come from the T cell precursor. The T cell precursor then go to the thymus, where they're gonna go through this process here, positive selection and negative selection. Remember, the positive selections happen in the cortex, and in the medulla, a little deeper, that's where it happened, the negative selection. In this process of negative selection, they divide in CD8 and CD4. They cannot have both at the same time. Now, remember, in the cortex, they used to have CD4 and CD8 together. But now, they divide in the CD8 and CD4, so they cannot have both at the same time. That happened in negative selection in the medulla. Then they come out of the thymus and they go to the lymph node. When they are in the lymph node, they divide again in 4 different groups: Th1, Th2, Th17, and T regulators. Remember, in the lymph node, they turn in CD8. It's called cytotoxic, and CD4 is T helper and cytotoxic T cell. So remember, the cytotoxic T cells divide in Th1 and Th2 by the effect of the interferon gamma and interleukin 12. Remember, interferon gamma and interleukin 12. This is very important. I have seen a lot of questions about this. So the other group on Th2, by the stimulus interleukin 2 and interleukin 4, they convert into the Th2. This graphic is very important. It's very good to learn it, and because I've seen a lot of questions about here, like which one stimulate the Th1 or which one control, like is, because you can see here the TDH is stimulated by interferon gamma and interleukin 12, but then it's blocked by interleukin or control or by interleukin 4 and interleukin 10. So I have seen a couple questions about that. As we remember, we talked about this before. It's the natural killer. They produce interferon no, they are stimulated by interferon alpha and gamma. They produce interferon. No, correction. I'm sorry. The macrophage are stimulated by interferon gamma, and they turn into a natural killer machine, the macrophage, by the effect of the interferon gamma. Remember the cytotoxic CD8 in the group of Th1, by effect of interferon gamma and interleukin 2, they activate the macrophage and cytotoxic T cell. They secrete interferon gamma and interleukin 12 and activate the macrophage. Something that I would like to clarify here is that this whole group of Th1 and Th2, Th17 and T regulator, they all come from TD helper. You can see here it's very confused because I was saying that it's come from cytotoxic T cell, like a CD8, cytotoxic, and then go like this. All of this come from CD8. All this group come from CD8 TD helper, CD8 helper. It's good to clarify this because I was confused too. So in this graphic, it doesn't show that anything come from cytotoxic T cells. So all this group of cells all of them come from uh, uh, T helper. This is very high GA because I've seen questions about which one stimulate and which other control the secretion of each one. So it's important to know which one is stimulate and which other one block. So I would say the most important here to learn from those one is that Th1, remember CD4 helper, Th1, they secretes alpha interferon gamma and interleukin 2, and they activate the macrophage. They activate the macrophage and cytotoxic cells. And the other group, Th2, by effect of they secrete interleukin 4, interleukin 5, and interleukin 6 and 10 and 13, and they activate eosinophils. So in all kind of allergies process, this is by the effect of Th2, because they activate eosinophils and interleukin E. Remember, they are elevated in allergies process and allergies reaction type 1. So Th17 then by the effect of interleukin 17 and 21 and 22, they induce neutrophilic infiltration. And remember, CD4 helper, by effect of the T regulator, they prevent immunity to maintain its self-tolerance. The most important here is high GST Th1, by the effect of interferon gamma and interleukin 2, activate the macrophages. So as we can read here, macrophagal lymphocytes interaction. Macrophagal lymphocytes interaction. So the Th1 cells secrete interferon gamma, which enhances the ability of monocytes and macrophages to kill microbes they ingest. The function of this is also enhanced by interaction of T cells and CD40L. This molecular, guys, you have to learn this molecular. This is very important. Let's see, CD40L. Very high here. So this function is also enhanced by interaction of T cells CD40L with CD40 or macrophages. Macrophages also activate lymphocytes via antigen presenting. So every time you see interaction between lymphocyte and macrophage is through this here, CD440 and CD40L. CD40 and CD40L is related. They're both related with the interaction of the macrophage and lymphocytes. They work together. They link together through this molecular here. So this is explaining kind of the things that we saw in the graphic before. So cytotoxic T cells, cytotoxic T cells kill virus-infected neoplastic and donor graft cells by inducing apoptosis. Release cytotoxic granules containing preformed proteins like the perforin and granzyme V, exactly like natural killers do. They contain or release perforin and granzyme B. Cytotoxic T cells have CD8 with bind to MHC1 on virus-infected cells. We know that before. So remember, CD8 bind to MHC1 and CD4 bind to MHC2. Important to remember those one: positive selection, negative selection, interaction of the macrophage and lymphocytes through the CD40L and CD40. The cytotoxic T cell, they act like the natural killer. They kill virus infected through perforin and gramzyme V. Then we're going to jump into the regulatory T cells. So regulatory T cells, let's see. They help to maintain a specific immune tolerance by suppressing CD4 and CD8 and T cell effector functions. So regulatory T cells, they help to maintain a specific immune tolerance by suppressing CD4 plus CD8 plus T cell effector functions. They identify by expressing CD3, CD4, and CD25 and FOXP3. They are activated regulatory T cells. In the graphic, we can see it like Treg or T regulatories. They produce anti-inflammatory cytokines like interleukin 10 and transformer growing factor beta. So, produce anti-inflammatory cytokines like transforming growing factor beta and interleukin 10. This is important because if you have these regulatory T cells, if you have immunodeficiency of this FOXP3, you're gonna develop what is the IPEX disease, the group of IPEX disease that are immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. Genetic deficiency of FOXP3, this one here. If you have a genetic deficiency of this FOXP3, you're going to have autoimmunity that is characterized by enteropathy, endocrinopathy, nail dystrophia, dermatitis, and or other autoimmune dermatology conditions, and it is associated with diabetes and male infants. It is associated with diabetics and male infants. So IPEX, let's find out about it. So here in Google said IPEX syndrome is an acronym for immune dysregulation, polyendocrinopathy, and enteropathy, and X-linked syndrome. It is a rare genetic disorder primarily infecting males, characterized by the dysfunction of the immune system that attack the body's own tissues. It is caused by mutation on the FOXP3 gene, which is crucial for the development and function of regulatory T cells, a type of immune cells that help to control the immune response. So what happens if you don't have the proper regulatory T cells, all those immune cells can mistakenly attack healthy tissues, leading to various immune disorders. Okay, so that's why this group of regulatory T cells are important. If you don't have this under control, you can have this disease called IPEX disease or IPEX syndrome: immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. So, immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome are the most common known as IPEX syndrome, and is a rare genetic disorder that can present early in life with diabetics, diarrhea, and eczema. Remember that. So in the next video, we're going to read about T and B cell activation. Thank you.